Hello everyone, my name is Ali and I'll be presenting today about investigating gene expression differences between a low metastatic group and a highly metastatic cell line in human breast cancer. And I'll be using GEO2R to analyze differentially expressed genes and see some insights about that. The motivation behind the study is asking ourselves whether we can detect common differentially expressed genes that are significant to metastasis, which is basically the spread of cancer, and particularly in the specific cell line, just for the limited scope and being more in depth into finding what kind of insights we could uncover from this. The study hypothesis will be formulated as follows. We're trying to understand differences in gene expression profile between cell lines. And basically, can we find common genes that correlate with MDA-MB-231 metastatic potential, like basically its potential to spread. Some background on cancer. Usually cancer is an abnormal cell growth that is uncontrollable and it invades body parts, and it's the second leading cause of death globally. Some characteristics: it divides uncontrollably, it hijacks normal cells, and it infiltrates normal healthy tissues, and it can spread throughout all the body. And different cancer types have like different spread potential, and basically some are more benign than others, and their spread is usually on a spectrum. Not all of them are um very very potent in spreading. And basically they come in different types. Carcinomas are they are cancers that grow within the epithelial cells, basically on the skin, or more like all the lining of the organs. And sarcomas usually develop in the bone and soft tissues, and myelomas usually target plasma cells in the bone marrow. Leukemias are cancers that form within blood cells. Lymphomas, cancers evolving from the immune system, and some mixed types. A brief overview of breast cancer here, because we're the specific cell line that we're talking about, and we'll discuss what is a cell line after. Um, is about breast cancer because that specific cell line is actually derived from breast cancer, and it's one of the most aggressive ones. So basically, breast cancer is the most prevalent cancer in the United States. It's a leading cause of mortality worldwide among women. It has a 5-year survival rate. I mean, within 5 years survival rate, the the rate approaches to 90%. But even though 30% of breast cancer patients ultimately die from their disease just because of its recurrence at distant sites, and that means it's due to spreading potential. So, out of those 90 that recover within 5 years, 30% of them will ultimately die because of metastasis. And it's more prevalent in middle aged and older individuals, where the median age of diagnosis is 62. So, what is metastasis? As we know, as we said before, it's the spread of cancer. And basically, it's cancer spreading to other parts of the body. Also known as stage 4 cancer, it's advanced. It's an advanced stage of cancer, and it becomes untreatable. And this is where, like, when they say like cancer becomes untreatable or uncontrollable. So basically, tumor forms, cancer um, evolves and tries to leave the primary site. It goes through the bloodstream, and then it like goes into other tissues. It spreads. Like this is a picture of like the liver. Goes through the bloodstream. It reinitiates its growth and grows a tumor there. And basically, metastasis usually like targets different kinds of like locations within the body. Especially for breast cancer, specifics were in the brain, lung, liver, and bone. So Basically, it's there's a lot of also processes involved. So usually their their survival within the bloodstream is also dependent on circulating blood, uh, blood pressure, adapting to the new environment, whether it's going to be allowed to be there or not, and their confrontation with the immune cells, obviously. So, what are cell lines? Cell lines are basically models for cancer, and they're hard to like establish. Um, but the ones that are established, they are actually grown in vitro within cell cultures, and basically they're used widely used in different fields of medical research, especially cancer and drug discovery. But why they are, if they are so if they have like some differences within in vivo, why they're so important, it's because their ability to provide indefinite source of biological material, which means they can grow indefinitely if the successful establishment of a cell culture proceeds. And basically within that, they still, as I was reading, they retain like 80% of cell lines, I would say, um, are are like 80% of the characteristics of cell cultured cancer cells mimic the true model of a cancer behavior. So cell line establishes like there's problem in establishing it, as we said, due to environmental differences, of course, because in a culture a cell cancer doesn't have all the nutrients and support or the blood vessels to behave like in terms of spreading and all that as it has it in the body in vivo or like in experimental mice or in humans, which it has all the other tissues and mechanisms to it like that are also at play. So overview of the selected cell lines in our study. In this study, we're looking at low metastatic group and high metastatic group. So we specifically are looking into this cell line because it's the widely used in TNBC research. This means triple negative breast cancer, and the name was derived. It's because of the hormone receptors that this type of cancer doesn't have. Its negative means it has absence of estrogen receptor, progesterone receptor, and the HER2 receptors. So these are receptors that are very important, which these two have, basically to express estrogen and other hormone receptors. This is very like necessary for test and trial of like hormonal therapies, which some types of cancer actually interact with very well, and that's why MTF7, for example, is widely used for studying breast cancer biology and testing drug responses. So metastatic potential for these two is low to none, which means they rarely spread or they are very low in spreading potential. This one is very high. Um, in terms of, oh, we forgot to say about shapes there, but in terms of shapes, they come in different shapes: round, mask, grape-like, stellite, and basically you could tell by the picture of why so. And these are characteristics of basically of those shapes. Um, our MDA-231 is stellate shape, and the MCF7 is a mass, and ZR is like grape-like. So these are their, as you can see here, like their shapes. Um, Data source and validation. So what did we do? Here we accessed GEO for a specific series that involves these cell lines, that has these cell lines as samples, and basically it's a study that did a transcriptome analysis of 15 different breast cancer cell lines, and the data can be accessed through this series by clicking on it. Here we follow the step for reproducibility. Basically, you click on the accession number, scroll down to the analyze with GEO2R, and then define our group, and so define the groups that you want. Controls go first, which is important here of what you want to contrast with. So this is the experimental, highly metastatic. This is the control, low metastatic, and then you select your samples as I here chose them, and you basically assign the group after selection. So why ZR71 is chosen as in the low metastatic group? Well, basically, the more you have samples, the more you would be going towards an average. I didn't want to choose just MCF7 because it doesn't model like itself. It's as a control for MD-231. So basically, I needed at least one other one, and I limited the other choices. These two choices that are also like estrogen receptor positive with the same kind, but I did not find clear metastatic status, whereas for ZR, usually they are grouped with MCF7 as low metastatic candidate. So that's why I chose them. And I chose ZR. And usually DEC library that GEO2R uses, which this is what performs differential expression analysis, strictly requires at least two samples per either group. So I didn't want to choose another in the experimental because I'm specifically looking at MDA-231, but here I was free to choose whatever meets the requirements of low metastatic candidate. But those two happen to be the ones. Uh, so basically, the series is limited in terms of I don't have replicates for MDA. I have other cell lines, but that then we will just be looking at all others versus all others. I just wanted to look at this one specifically, as I said before. And that's why more samples per group in our study is bad. That's why I stated that here, because then I'm deviating from this specificity here. So the online resources used is GEO2R, as I stated before, from the National Library of Medicine. I'm comparing two or more sample groups to identify genes that are differentially expressed. And based on the supplemental material that I checked here by downloading this Excel sheet, I found out they are using FPKM, which is basically a normalization method for like that reads for fragment per kilobase millions. So they're normalizing per fragment here. And in the options menu, the Benjamin Hochberg p-value adjustment was used for false discovery rate, and a p-value cutoff of 0.05, which is basically the norm, could have went down, but when I tried zero point 0.1, I got around 800 significant genes, but with this I got a bit more, around 1200. We'll see after. Here, data processing. So after we said that we define our groups, control first, then highlighted the cell lines and assigned the groups, under the GEO2R tab, I clicked Analyze. And after that, I had findings. The table shows up with a lot of information that is useful, but the most useful ones to look at are the adjusted p-value because this one is more significant than this column because it accounts for multiple testing, which is more reliable. It takes into account all the other observations, all the other samples. So there's an option to download these, and it's 250 genes are displayed first, but results are presented in a table in the order of significance, obviously. Um, in the next one, I actually there's also besides the table, there's also like other interesting plots that there's that the software gives, and basically we found around 1262, and basically those were significantly dysregulated, which means they were up or down the genes that were found, and under this adjustment, of course, and basically out of 21,206, these were the most significant, and their distribution of p-adjusted value goes in ranges. So most of them are not that significant, but here's this peak here that is below zero point 0.5, which is of interest. And if we proceed to look at the other, which is okay, the volcano plot is basically the the main plot that was continued from here in terms of analyzing what it means. Um, Here are the up and down regulated genes, down in blue, up in red, and all of these colored genes are basically falling within the 0.05 p adjusted value. So basically, they're all considered significant. The transformations here, don't get like frustrated about what's negative log 10 and log 2 fold basically are okay. P-values are very, very small. And basically, to be able to discern, if you plot them without transforming the values, they'll be like on top of each other. You couldn't see like their spread in terms of like how they look on the graph. So they use transformations like negative log 10 is a standard, and they apply to the p-values, and basically it extends them over the graph to be able to see. In contrast here, so oh, not in contrast, still like with the p-value adjustments. So basically this line I drew here is basically a 1.3, and it is where the negative log 10 happens to be zero point zero 0.05, and that's why these are like gray, these are colored, and that's the separation of significance. So in contrast here, the log to fold change actually is also a transformation, and it used to make the data easier to visualize. And all what it means, I highlighted these points. If I just took into account this point, anything above zero is considered upregulated, anything below is considered down. So I highlighted this specifically to show you that if I project it down, I would get a 10 here. So if I put log to 10, its interpretation would be that this specific gene is tenfold expressed. And basically, if you want to understand it in terms of the opposite, well, the opposite of logarithm is exponential. So 2 to the 10 is this much. So this gene is 1,024 times higher, displays higher expression of that much than the reference example. And basically, for the down one, it's like 1 over. So it's like off the reference. So what are we doing here? So here we got the results, and right away I saw like regions of interest. These genes are relatively more spread in terms of significance, so because they're very high on the Y value. And these ones, for example, are very upregulated on the extreme. These are still a lot of genes to look at, but even though there are a lot, even those ones below them are of significance. So there's a lot even to look at. But what I did here is, first I selected columns here for this table because I want to include, you're able to include like other columns that are not displayed right away. So I actually activated them, and after that I was able to download all the genes. Given this feature here, you press on it, you get, by the way, you get the 21,000, all of them. And when you get all of them, why did I take this table? Is because it has more information. As I said, this is interesting and needed. We will see like how we'll use the process later. Um, but this is just for more information that we will show you later on what I mean. But in order to look at the significant file genes, we don't know how, we don't download only the significant file. So why don't we just download the significant file? Of course, I just downloaded the all genes before. Here, the significant, if I click on the volcano plot and explore the data and download option, there's an option to download just the 1200 genes, around 1200 genes that are significant. But basically, this dataset will include just these columns. They are of interest. The total number of genes is of interest, and there's some interesting stuff, but we also missed like the process, related stuff, and function. So what I did is I took those two files, all the gene file and significant gene files, and basically they're in TSV, which means tab separated. So they're just basically tab separation. I plugged them into RStudio. I imported the data and did some manipulation, trying to like look up significant, extract, and reformulate the data set, so I'll be able to have more information and segregate those specific regions. Because I tried to limit it even further because those are the regions that I was interested in. And basically, based on this criteria, I was able to look, basically, for example, the one here, it's at negative, around negative 13. So if I take log folds less than negative 13, I'll be able to filter that group. and same goes for the others. I was able to filter them. Um, this is all this criteria was implemented in a code, and basically the code will be shared on GitHub through a link um, afterwards. And the process ended up with this. So the genes of interest happen to be here. We have the table of most significantly downregulated because they're at the extreme. They're not more than this significant, but all of them are significant, obviously, but they're the most downregulated. This one is the most downregulated here, the four, two, two genes, you see them, and the most significantly downregulated, significantly because they're above. This one is the most downregulated because they're the most right downregulated, but they're not significant as those. Three is the most significantly upregulated. So basically, you see my point here. But I got a list of very important genes that could be looked at, and like they're very much of like interest. For example, this specific gene exists is of recent interest to my partner, which is doing her PhD in like molecular and cellular biology, and she's specifically looking at MDA-231. So at some point, she realized like, okay, this data aligns with the significant status of exist. So interpretations here. Basically, the model was the results were aligning with the all significant genes here. So all of them here are aligning with the all significant genes file, and everything was imported and demonstrated as shown before. But what I did further for understanding some of the genes is I looked into a paper which had a model system based on 231 cells and has a rigorous vitro and in vivo study. It explored a lot of genes, but I was able to just pick some of them to see what I could get. Um, and basically, they grouped like their metastatic status based on whether a gene initiates or like has aggressive spread, or it suppresses, or it's like anti-spread behavior. So the categorization of these genes, I just kept them as is within their group, but I also looked and try to search whether I find those genes within my file, and basically all the significant genes file, not all the genes. So basically these are the 1260 something genes. So I realized that the ones that are colored were found inside. This one I'm not sure, they had a typo in the name, so I didn't know if that's another gene. I looked it up, I didn't find, but this was inside my list. So this is a common gene known here. So basically I found these four, and the color that I have here indicates whether they were upregulated or down based on my data. So basically here, interesting, when I looked up those genes, I was able to look at my data set, and remember the column that was of interest, the Go process column was able to give me this information. So basically, these two genes are involved in angiogenesis, which is basically blood vessel development, develops new blood vessels for tumor. And since this is downregulated, I was surprised because this is a promoter gene and it's downregulated. Similarly, this other gene is a negative regulator of angiogenesis, which has the opposite behavior of these two genes, but it was upregulated. It's a suppressor gene that is upregulated. That's another surprise here. And then I realized this one also is a suppressor gene. It basically regulates blood pressure, which could help in the transport of cancer cells to different sites. And it's also upregulated. So what I found out that the functions aligned with the study. The functions of these genes aligned with the study, but the regulation was the total opposite. So I was like in kind of a paradox here. So only four genes were found to match the list of the genes derived. So basically the ones in color here, I mean the four genes. And in terms of dysregulation, genes from the study did not align with the results from GEO2R. cross-reference with the Go process column. So that got me to think, well, okay, I'm dealing here with a cell culture, and basically the environment is different, and metastasis behavior could be different entirely because there's no blood vessels formation and all these kind of stuff. So that could have explained little why such behavior exists in my results based on cell line. So I ended up concluding that MDA-MB-231 cell line, known for its high metastatic potential, may utilize different genes and pathway for angiogenesis, which is blood vessel formation, and blood pressure regulation, compared to less metastatic cell lines like these two. So in a sense, even I'm doing this differential expression specifically to 231, if you reverse the experiment, this means the gene regulations will be different, will be the opposite, and it will reflect the status of these two, which basically is interesting. So the limitations used in this study is that we only use one sample in this experimental group. We could have used more if there was. These two were used to model non-to-low behavior, so these two controls were used as a control. Maybe there's other cell lines that are a better candidate for controls. And this study only referenced the set of genes from one study. So basically, this lets me to say that for future suggestion, we could be looking at an increased sample size for MDA-MB-231 to validate the findings and prove the statistical significance and reliability of the genes we find. We could also have functional study to conduct functional assays to like look into the impact of genes in metastasis, especially to those that are specific to MDA, so more experimental designs. Or we could just do pathway analysis and perform like an in-depth and look into like angiogenesis, like the findings we just got, like why such behavior exists, and in terms of like blood vessel formation and blood vessel and blood pressure regulation mechanisms in this specific cell line. Um, or also we could cross-reference more studies to find more comprehensive set of literature that uncovers more genes that are involved, which basically, okay, don't rely just on this one specific study with all its genes, although it's a comprehensive study, but there might be other studies that could provide more insight into specific genes that are more in depth studied that are related to metastasis of this specific cell line. And this is a list of references that I've used throughout my presentation, and I look forward for your suggestions on the discussion board. And thank you so much for listening and um, following through. Thank you.